third lab of the semester is the histology lab. Histology is the study of tissues, particularly at the microscopic level. So this slide, if you were taking it in person, would be the one where you look at a whole bunch of microscope slides. Of course, we don't have that option unless you go into Open Lab, but luckily we have pictures of each type of slide included in our lab manual. So I'm going to skip past this table. This is not part of the lab experiment or anything. This is just kind of for your reference. So our first set of slides are epithelial tissues. This lab will make a lot more sense if you've done the tissues lecture before you do this lab. The first type of epithelium that we have is the simple squamous epithelium. So it's simple because there's one layer of cells. It's squamous because those cells are flat. So what we're looking at here is this is a lung. This is a slide from a lung. And so each of these lines are cells. It's cell next to another cell lined up in a line. If you look at it, you can kind of see that there are some darker spots. Those darker spots are the nuclei. The lighter area between the nuclei is the cytoplasm. And so there is a single layer of cell going through each of these lines. So this is unique. This is the only only slide that's going to look like this. And a simple squamous epithelium is usually going to look very, very similar to this slide. So make sure you go through and you write where simple squamous epithelia can be found. This information is in your book. It's also in our lecture. Next is the simple cuboidal epithelium. So it's simple because there's one layer of cells, and it's cuboidal because that single layer of cells is a cuboidal shape. They look like cubes or spheres. They're essentially the same width as they are height. And so if we look, we can see there's one cell next to another, next to another, next to another, next to another, next to another. It's one layer thick. Again, dark spots are nuclei. That's what I would suggest when you look at a slide. Look for the nuclei. That'll tell you where the cells are, and then look and see what is around the nuclei. And so these are kind of block-shaped cells. I hope you can see that this is different than this, the simple squamous epithelium. These are flat. These are cuboidal shaped. Make sure you write down the locations. Simple columnar are simple because it's one cell next to another, next to another, next to another. They're columnar because they look like columns. They're tall and skinny. As always, the dark spots are nuclei. There's 1, 2, 3, 4, 5, 6. They're stacked next to each other, and they are tall and skinny. It wants you to identify a goblet cell. A goblet cell is named a goblet cell because it looks like a wine glass or a goblet. And so that is these large areas here. And so this is the cup part of the wine glass. And then if you look where it meets here, some of them will have a stem. You can see a cup and a stem there. The goblet cells make mucus, and then they dump that mucus through the stem out into this open area here. So those are our three types of simple epithelia. Next we have the stratified epithelia. Remember that stratified means there are multiple layers of cells. And so it'll be called stratified something epithelium. The shape refers only to the outermost layer of cells. The outermost layer can be a different shape than the underlying layers. This shape in the name only refers to the outermost layer. So this first slide is the stratified, squamous, non-keratinized epithelium. So we're going to work our way through this. It's stratified because there are clearly a bunch of layers here. So you can see where we go from pink to purple. The purple area here is the epithelium. That's what we're looking at here. It's stratified because we have cell on top of cell on top of cell. It's squamous because the outermost layer is flat. The ones at the bottom aren't, but that doesn't matter. The outermost layer is flat, and so it's squamous. This is non-keratinized as opposed to keratinized. And so if I scroll down a little bit so you can see them both, I hope you see that for the most part, this area here looks like this area here. But this one has this extra layer of milky looking fibers up here. That is keratin. Because there's keratin up here, this is called keratinized. This one is non-keratinized because it does not have keratin. Keratin forms kind of a hard protective layer or shell on the outside of this epithelium. Got some questions for you to answer. Then we move on to connective tissue. And these look kind of unique. So the first one here is areolar connective tissue. Areolar and adipose are considered loose connective tissue because if you look at them, there's a lot of open space in them. We'll see in the dense connective tissue, there is not. So in the areolar, there's a lot of things going on here. So it wants us to label fibroblasts first. Fibroblasts are cells. Look for nuclei, dark purple spots. Those are the fibroblasts. Fibroblasts make fibers. It's a nice, easy to remember name. Next, we have elastic fibers and collagen fibers. So I hope when you see this, you can see that there are large fibers. They're kind of pinkish colored, and then there are small, skinny, purple fibers. The collagen fibers are the large ones. The elastic fibers are the small ones. You won't see another type of tissue that looks like this. Areolar connective tissue is common in our body, but it has a very unique appearance to it. Next is adipose tissue, which is fat tissue. And so what you're looking at here, it looks like there's a lot of completely empty space. It's not just empty space, though. Each of these circle areas is an adipocyte. That is a fat cell. And then you see the purple dots, as always, are the nuclei. In each adipocyte would be fat. So this is a microscope slide. It was sliced. When they sliced it, the fat came out. So on the slide, it is empty space. But in your body, it is filled with fat and lipids. Next is dense connective tissue, where everything is packed in very close. So this is dense regular versus dense irregular. Regular is lined up in a line. It's very neatly packed. Irregular is kind of randomly laid down. Regular has fibroblasts. Fibroblasts are cells. Look for the purple dots. Those are your fibroblasts. The purple dots are the nuclei of the fibroblasts. And then all of this large pink is collagen. Dense irregular is essentially the same thing. The pink is the collagen, but the fibers don't run in a single direction. Dense irregular connective tissue is good for stretching many different directions pretty well. Dense regular can only stretch in one direction. So this can only stretch in a kind of diagonal here. It would not be good for stretching this direction. But the direction it's made to stretch, it stretches really, really well. So this is going to be found in something like a tendon, where it's designed to stretch one way, whereas this will be found in other parts of your body, where it's going to stretch in multiple different directions. And finally, we have blood. Remember that blood is considered a connective tissue. That's hard to remember. That's easy to forget. It is a fluid connective tissue. It's difficult to think of it as a connective tissue because it doesn't really connect one thing to another. This is the same slide we saw on the cytology lab. And so these pink cells are the red blood cells. The things with the purple, where you're seeing the nuclei, the DNA that has been stained, that's the white blood cells. The area around it would be the plasma, which is the ground substance of this connective tissue, or the extracellular matrix. Some questions for you, and you can fill in the location and function of these tissues. Let me know if you have any questions, and when you're ready, you can move on to the next lab.